Good afternoon, everyone, and thank you for joining us for the latest installment of the HDSA Research Webinar Series. This is George Orling from the HDSA offices in New York, and I'm excited to welcome our speaker for our first speaker of the fall, Dr. Helen Budworth. But before I introduce her, for those who are new or has been a while since they participated in a webinar, I just wanted to remind you about how you can ask questions. Alongside, on the right-hand side of your screen, you should see a GoToWebinar menu, which can open up and look like this, where you can expand the questions box and type your question at any point during Helen's presentation, and just click send. And at the end of Helen's presentation, we'll be reading those to Helen on your behalf. So as soon as a question comes to mind, feel free to submit it, and we'll do our best, if time allows, to get it answered. We recognize that, as always, at these research webinars, we try to accommodate as many people as possible. That it might be a little early for folks on the West Coast, particularly Helen, which we appreciate her joining us at such an early morning hour. But we are recording this, and we'll archive this on the HDSA website, as well as HDSA's new YouTube channel, so people can view this and share it with family members or other community members as they see fit. And I'll just remind everyone that we have another research webinar coming up in October 7th at 12 p.m. Eastern. This will be presented by Dr. Christina Bekanovich from the Karolinska Institute in Sweden, where she'll be presenting her work in a recent paper that was involving a DNA variation, or a SNP, a mutation that was found in the Huntingtin promoter that appears to be a genetic modifier delaying or accelerating that disease onset of Huntington's disease. So stay tuned for that on October 7th. And as always, if you have a research webinar topic or an area of HD research that you think is very exciting and HDSA hasn't covered and you'd like to hear more about it, please send those suggestions to researchupdates@hdsa.org, and we'll make sure that we'll do our best to try to find an applicable speaker to cover that topic for you. So I just want to turn it over to Helen Budworth. Dr. Budworth is currently a research scientist in the Genome Dynamics Department and Life Sciences Division at the Lawrence Berkeley National Laboratory in Berkeley, California. She's also, I'm really pleased to say, one of our first recipients, our 2013 recipient of our HDSA Human Biology Project Fellowship, where we're funding her to identify metabolic biomarkers in HD patients' plasma. Before joining Lawrence Berkeley, Dr. Budworth received her bachelor's degree at the University of Liverpool in England, and her PhD from the University of Oxford, and joined as a postdoc at Lawrence Berkeley in 2004, where she's been ever since. Today you're going to hear a little bit about some exciting data coming from both Helen and her lab at Berkeley regarding the suppression of somatic expansion. This is the expansion of the CAG tract in Huntington's disease. When it expands, causes the disease. They've done some really exciting work in mice, HD mice, both using genetic crosses as well as a pharmacological agent to suppress the expansion of the CAG tract. And you're going to hear about this exciting data from Helen right now. So I'm going to turn it over to you, Helen.
Okay.
The floor is yours.
Okay, thank you, George. Thank you very much for that introduction. It's a real pleasure to have this opportunity to present our research for the HDSA webinar series. I'm very happy to share the results of our recent work. So, as George mentioned, today I'm going to talk about our recent work, the suppression of somatic expansion in a mouse model of Huntington's disease. So I'll just run through an overview of the talk. I will give an introduction to the genetics of Huntington's disease. I will then talk about expansion of the CAG repeats that goes on in cells of the brain. I will discuss DNA damage, DNA repair, and how that relates to Huntington's disease. I will introduce our mouse model of Huntington's that we used in these studies, and then I'll go on to describe the studies themselves and the results that we found. So the work that I will talk about today is from a manuscript that was recently published by our group in August of this year in the journal PLOS Genetics. This is an open access article, so I encourage you all to visit the PLOS Genetics website to read the full manuscript. So to give an overview of the genetics of Huntington's disease, the Huntingtin gene is found on the short arm of chromosome 4, which is highlighted here in orange. HD is an autosomal dominant condition, which means that inheritance of a single copy of the mutated expanded gene is sufficient to cause the disease. Therefore, if one parent carries the Huntington's mutation, there is a 50-50 chance that each child will inherit the mutation. The mutation is caused by expansion of a repetitive stretch of CAG repeats. The C is cytosine, A for adenine, and G for guanine in the DNA, and this triplet repeat is found in the coding region of the Huntingtin gene. In normal cases, this CAG triplet is repeated around 10 to 30 times within the gene, but in Huntington's disease, this CAG stretch expands and is repeated more than 36 times. People in the intermediate range with around 30 to 37 repeats may or may not develop signs and symptoms of HD within their lifetime, but they are at risk of passing the condition down to their children. This CAG stretch undergoes expansion between transmission from parent to child at fertilization, and children of Huntington's patients tend to have a larger number of CAG repeats than their parent, and this larger number of repeats is associated with earlier onset of symptoms. The mutant Huntingtin protein is shown here. An increase in the size of this CAG repeat leads to the production of an abnormally long version of the Huntingtin protein. A series of 3 bases in DNA is known as a codon and codes for a specific amino acid in proteins. In the case of the CAG, the amino acid that this codes for is glutamine, which is represented by the letter Q. Therefore, a series of repeating CAGs produces a polyglutamine or a poly-Q tract. This long glutamine tract is very sticky and leads to the formation of protein aggregates, which is one of the problems that you get with the Huntingtin mutation. Huntington's disease is a neurodegenerative condition. The mutant protein aggregates and accumulates in neurons, which is a cell type within the brain. Additionally, this elongated protein can be cut into smaller fragments that are toxic, and these accumulate within cells of the brain. These protein accumulations can disrupt normal neuronal cell function and lead to cell death of neurons in the brain. And this dysfunction and eventual death of neurons in certain regions of the brain are the hallmark signs and symptoms of HD. What I'm showing here on the left are the particular regions of the brain, and on the right at the top is an image of a Huntingtin brain, at the bottom an image of an unaffected brain, and you can see the difference in size and the areas of cell loss in Huntington's compared to an unaffected brain. So the mutant protein causes death of brain cells in a region-specific manner. It specifically affects regions of the brain that control movement and leads to motor decline and cognitive impairment. The most prominent cell loss occurs in the forebrain in the striatum and the cerebral cortex, in which certain cells known as medium spiny neurons and cortical pyramidal neurons are most affected. There is currently no cure for these symptoms and signs of HD or treatments that modify this disease progression, so therapies are very much in need. Huntington's appears generally in most people later in life, and this graph that I'm showing here shows the relationship between the number of CAG repeats in the DNA of Huntington's patients and the age of onset of disease symptoms, which could be involuntary movements, cognitive or psychiatric abnormalities, or the inability to perform complex hand movements. As you can see, the number of CAG repeats along the bottom or x-axis, as this increases, the age of onset, which is on the y-axis on the left, decreases, so that a person with more than 70 repeats, as you can see on the bottom, will likely get onset of symptoms before age 20. The red line that I'm showing on this graph indicates the position of 44 CAG repeats on the x-axis. If you look at the y-axis for age of onset for a person that inherits 44 CAG repeats, you could see onset of disease as early as age 20 or as late as age 85. But the average age of onset for a person inheriting 44 CAG repeats would be around age 50, and there's wide variation in this. So this suggests that onset of the disease is not only dictated by inheritance of the expanded gene, there are other factors involved with onset of symptoms. Somatic expansion is expansion of CAG repeats in cells of the body known as somatic cells. That is any cell of the body that's not a germ cell, a sperm or an egg, and not an undifferentiated stem cell. And this expansion goes on in cells throughout the body throughout life. Somatic expansion has been previously suggested to contribute to onset of Huntington's disease, but this has been very difficult to test. Effects of Huntington's manifest in the brain and cannot be followed in humans throughout life. Analysis of HD patient brains post-mortem has suggested that somatic expansion could occur in Huntington's disease, but the cells that did undergo expansion are likely to be cells that were lost due to the toxic protein that was produced. Also, all Huntington's patients inherit the expanded mutant gene from a parent, and this alone leads to development of Huntington's. So trying to assess the effects of any further expansion on top of what was inherited are very difficult to separate from effects of just the inherited gene. What I'm showing here is a schematic that depicts alternative models for Huntington's onset at age 50. Across the top is the CAG repeat length, and in the conventional model of Huntington's, inheritance of a gene containing 42 repeats at birth could lead to decades of effects from the protein toxicity of this expanded gene, and onset of symptoms would occur at age 50. On the bottom, in an alternative model, inheritance of an expanded gene with 42 repeats leads to instability of that repeat region in somatic cells, which causes somatic expansion of the CAG tract up to a threshold number of repeats, at which the patient would show onset of symptoms. And this somatic expansion model would open the door for potential therapies to stabilize these repeats and delay reaching the threshold at which symptoms could appear. So how is DNA damage and DNA repair involved in Huntington's disease? Expansion of the CAG repeats occurs through a process of DNA damage and attempted DNA repair. Oxidative damage happens all the time in all cells. It's an inevitable consequence of our cellular metabolism. The mitochondria, which are the powerhouses in the cell, are major sites of reactive oxygen species or free radical production in the cell. These reactive oxygen species that are produced by the mitochondria can damage DNA. Oxidative damage to DNA occurs frequently at guanine bases, which is the G in the CAG repeats, as this base has a high oxidation potential, but oxidative damage can occur at other bases in DNA also. Oxidized bases are removed from DNA by enzymes in the base excision repair pathway of DNA repair. What I'm showing here is a schematic of the base excision repair pathway operating at CAG repeats. The enzyme shown in the blue blob is OGG1 or OG1. This enzyme recognizes and removes an oxidized guanine from DNA. Partner proteins in the base excision repair pathway then break the DNA strand that contained that damage and creates a strand break on one strand of the DNA. This broken strand allows another enzyme, a DNA polymerase, which is an enzyme that copies DNA, to come in and fill in the gap with the correct base, in this case, a normal guanine. And it does this by using the opposite strand of DNA as a template to know what base was missing. In the case of a repetitive CAG repeat region, it's been shown that this DNA polymerase gap-filling enzyme can slip as it tries to copy this highly repetitive region. Instead of filling in the single base gap, it continues copying the template strand, and as it does so, it displaces the adjacent strand of DNA. This slipping allows the formation of DNA hairpins. DNA hairpins, shown on the top right, are structures that form in DNA when single strands of DNA containing bases that can pair with each other. In this case, the C can pair with a G, and the G can pair with a C, and what you would have in the middle were two A's that do not pair. So the CG pairs are depicted in black with the black lines, and the AA mismatches are shown by the red dot. These hairpins are stabilized by the bases binding to each other, and then the repair process from the broken DNA at the beginning is completed when the ends of the break are stuck back together, and now the DNA on one strand has a hairpin containing CAG repeats. DNA hairpins are very hard for the cell to repair, and they can persist in the DNA as stable structures. When the opposite DNA strand is nicked or broken due to DNA repair on that strand or some other DNA transaction, this allows opening of the hairpin, and the extra CAGs are incorporated into the DNA, which is how you get the expansion mutation at the CAG repeats. To introduce our mouse model of Huntington's disease, we used the HDHQ150 mouse model of Huntington's. This is a knock-in mouse with 150 CAG repeats knocked into the Huntingtin gene. HDH stands for Huntington's disease homolog. The Q stands for glutamine, the codon coded for by CAG, and 150 is the number of CAG repeats or glutamines knocked into the mouse Huntingtin gene. HDHQ150/Q150 indicates that both copies of the Huntingtin gene contain the expansion mutation. For comparison, we also used an unaffected or wild-type mouse in the study, which is depicted by the letters WT, and a mouse with only one copy of the expanded Huntingtin gene, which is HDHQ150/WT. Since the Huntingtin mutation is dominant, this mouse with only one copy will also show symptoms of Huntington's disease. This is a late onset model, so it's well suited to analysis of the onset of symptoms, and symptoms that we monitor in these mice include loss of motor function and coordination, and these symptoms generally start to appear around 10 to 20 weeks of age in these mice. On the right, the OGG1 mouse is a knockout mouse in which the OGG1 has been knocked out or removed. The -- in brackets indicates that both copies of the OGG1 have been removed. Again, for comparison, we used a normal or wild-type mouse, as shown by the ++, and a mouse that has lost only one copy of the OGG1 gene, which would be labeled as +-, and this mouse would be expected to display some effects of having lost one copy of the OGG1 gene. This OGG1 knockout mouse has been studied in the past. OGG1 is not essential for life. By not recognizing and removing the oxidized guanine bases, you don't cause a break in the DNA, which will not allow the formation of the hairpins that lead to the expansion. You will accumulate oxidized guanine bases, but up to a certain level, these can be tolerated by the cell. So by breeding the Huntington's mouse with the OGG1 knockout mouse, we're able to study the effects of somatic expansion that will be present in the mouse with OGG1, but absent in the mouse without OGG1. So the questions that we wanted to answer with this study, the goals of the study were to determine if somatic expansion of CAG repeats did contribute to onset of disease, and if so, whether we could block somatic expansion and delay onset of symptoms in a Huntington's mouse. In order to do this, we took the Huntington's mouse with the OGG1 gene. This mouse will inherit the expanded CAG tract of 150 CAG repeats, and it will undergo somatic expansion due to attempted DNA repair by OGG1 that allows formation of hairpins and allows the expansion. We compared this to a Huntington's mouse without OGG1. So in this case, the DNA is not broken at the oxidized guanine bases, you don't get formation of hairpins, and you don't get expansion of the CAG repeats. We took groups of mice, ages from 5 weeks all the way up to 120 weeks. These mice were tested for motor function. We also collected DNA from these mice to test the DNA for the CAG repeat expansion. And then we were able to compare the effects with and without somatic expansion at various ages. What I'm showing here is a validation that the loss of the OGG1 protein does not alter any of the physiological properties of the mice. On the left, we looked at the litter sizes from the mouse breeding, and as you can see over on the left at the top, the Huntington's mouse with OGG1 has a litter size of around 6. The same Huntington's mice without OGG1, different group, also has a litter size of around 6. And these were similar to mice unaffected by Huntington's, the wild-type mouse, with and without OGG1. We also compared weights of the mice. The average weight at 25 weeks was taken, and what we saw no major differences in weights of these mice, although perhaps you will notice that the Huntington's mice over on the left did weigh slightly less than the mice without Huntington's. The Huntington's mice with OGG1 were around 25 grams, and the non-Huntington's mice with OGG1 were around 30 grams. On the right, I'm showing histological analysis of regions of the brain in the early ages of these mice. We looked at brains, and we used various stains that allowed us to look at tissue structure, and we used a stain that allowed us to count the number of neurons, and we saw no differences, no major differences across all the groups of mice at these early ages. The number of neurons were in a similar range. So this told us that the OGG mouse was a valid comparison to the knockout mouse. In order to measure the number of CAG repeats in the DNA, we took DNA from various tissues in the mice, including the tail and different regions of the brain. The region of DNA containing CAG repeats was amplified by the polymerase chain reaction by PCR, which makes many copies of each CAG region. The DNA from each mouse was then separated by size in a gel, shown here as an example DNA gel. In these gels, shorter DNAs move faster through the gel. Therefore, you get banding patterns of DNA that is separated by size. The lower band that you see, pointed to by the lower arrow, this is the normal non-expanded Huntington's allele, and this moves faster through the gel. The upper band, the mutant expanded HGHQ150 allele, moves slower. So in the red box, these are mice that contain 2 copies of the expanded allele, so you only see one band in these mice, and it runs at the slower rate. In the blue box, mice 9 and 10, these mice have 1 copy of the expanded Huntington gene and 1 copy of the normal allele, so you see 2 different bands in the DNA gel. In the green box, mouse number 17, this is a wild-type mouse, so you only see the faster-running, lower, normal Huntington's gene. Over on the right, you will see a spread of DNA from each band of the gel. So each band of the gel contains DNA with a mixture of lengths. So what I'm showing here in blue, you see the spread of sizes of DNA in the CAG region of the Huntington gene. On the top, you have the Huntington mouse with OGG1 that will undergo somatic expansion. So if you notice that in the brain, the blue peak in the brain is shifted to the right, which indicates that the CAG tract in this tissue is longer compared to the tail or the liver. In the middle panel, you have DNA from a Huntington's mouse with only one copy of the OGG1 gene. This shows a similar pattern to the mouse at the top, although you notice the spread of DNAs in the brain are somewhat shorter than in the mouse with 2 copies of OGG1 at the top. In the bottom panel, what you have is a Huntington's mouse without OGG1, so we are expecting no somatic expansion in these mice. And what you see is that expansion is less, and the spread of sizes of DNA in this brain tissue has not shifted as far to the right. So what we see is that loss of OGG1 suppresses somatic expansion. At the top, I'm showing a schematic of what we're seeing in the results of this study. A mouse with the Huntington's gene in the presence of OGG1 that will undergo somatic expansion inherits an expanded allele, and this expanded allele undergoes expansion throughout life to a longer CAG stretch. On the bottom, you have a Huntington's mouse where we've knocked out OGG1, so we don't break the DNA to repair the oxidized bases, we don't undergo somatic expansion. So in this mouse, the expanded HD allele is stabilized and does not undergo somatic expansion. To further highlight this, the graphs on the bottom of this slide show the range of CAG tracts in DNA from the striatum in the brain from a group of mice at 10 weeks of age. On the left, you have a group of Huntington's mice that contain the OGG1 gene and will undergo somatic expansion. Each color in the graph represents a different mouse, and each mouse has a range of CAG repeats in their DNA. This graph shows the change in the number of repeats over what they inherited at birth, which we measured in their tail DNA at birth. You can see that the CAG region in these mice is unstable, and some mice have an increase of up to 40 CAG repeats by 10 weeks. On the right is a group of Huntington's mouse where we knocked out the OGG1 gene. DNA from the brain region of these mice at the same age shows much less change in the number of CAG repeats. So this shows that instability of the CAG repeats is affected by OGG1, and when we knock out this gene, we stabilize the CAG region. We tested our groups of mice up to age 120 weeks, and we showed consistently that mice lacking the OGG1 gene had more stable CAG repeats, particularly at the early ages. So we know that knocking out OGG1 affects the DNA of the mice and stabilizes that CAG region. We also wanted to test whether it had effects on other symptoms in the mice. So to do this, we did motor testing of our mice. Huntington's symptoms manifest as cognitive defects and motor dysfunction, and these motor deficits can be tested in mouse models, and these are relevant to the human state. So in order to do this, we use a variety of motor tests that include the Rotarod test, where mice are allowed to walk on a slowly rotating rod. The amount of time that the animals stay on the rod is determined by a built-in magnetic trip switch, which is stopped when the animals fall off. So we time our animals on the Rotarod test. Also, mice were tested for their ability to grip onto a bar in the bar grasp test. The mice are lowered onto a bar and allowed to grip with their forelimbs. This bar is suspended over a padded surface, and the mice are allowed up to a maximum of 30 seconds to see if they can hold on to the bar. What we found was that suppression of somatic expansion and loss of OGG1 delayed motor decline in our mice. The graphs shown here show the time in seconds that the animals remained on the rotating rod, with 120 seconds on the y-axis on the vertical being the maximum allowed time. So as you can see, each dot represents a mouse. The black dots are mice without OGG1, where we knocked out the OGG1 gene. The white dots represent Huntington's mice with OGG1. And what you can see is that the results were very variable, as would be expected with groups of mice, and this also mimics the results you see in Huntington's patients. They have a variety of spectrum of symptoms. But we averaged our results, and what we could see was the trend that when you lose OGG1 in the solid line, you delay loss of motor symptoms. So the black line, solid black line, remains higher than the dotted line. And this is the case in an animal that has 2 copies of the expanded gene, which is what I'm showing here on the top, and the same in an animal that has only 1 copy of the Huntington gene. We see the same pattern, that by knocking out OGG1, we delay loss of motor symptoms. So what we believe is happening in these mice is that there is a toxic oxidation cycle. Here in number 1, you see the mutant Huntingtin protein. This has effects on the mitochondria in the cell. Mitochondrial dysfunction has been described by several groups as a symptom of Huntington's in cells of Huntington's patients. So the effects of the mutant Huntingtin protein on the mitochondria cause an increase in the production of reactive oxygen species, or free radicals. These free radicals damage DNA, so you get oxidization of bases at the CAG repeat region. The cell tries to repair these oxidized bases using the OGG1 gene to start the base excision repair pathway. By removing this oxidized guanine, it causes a break in the DNA. This break in the DNA allows slippage and formation of hairpins that are stabilized in the cell. These hairpins eventually relax and cause the expansion mutation, and this is a toxic cycle. So what we show there at number 3 is that in mice, by knocking out the OGG1 gene, we're able to block this cycle, reduce somatic expansion, and delay onset of symptoms in the mice. So what we also wanted to see is whether there were things that we could do in patients, whether we could pharmacologically suppress this somatic expansion and delay onset of symptoms. So to do this, we use a compound known as XJB5131. XJB5131 is an antioxidant therapy that is targeted directly to the mitochondria. So this compound is derived from an antibiotic, and it's targeted directly to mitochondria, and it contains free radical scavenging ability. Our group has previously reported on this compound for its ability to suppress oxidative damage and delay motor decline in a mouse model of Huntington's disease. In this study, we treated our mice with XJB, and we show that XJB also suppresses somatic expansion and delays motor decline in the mice. What I'm showing on the top left are Huntington's mice. Now these mice do have the OGG1 gene. This is a separate study just looking at the effects of XJB. We took DNA from these Huntington's mice from the brain at age 21 to 30 weeks, and we compared mice treated with XJB to untreated. These mice have 2 copies of the expanded Huntingtin allele. So in their blue peak profiles of the range of DNA repeats, you see 2 peaks which represent the 2 expanded copies of the Huntingtin gene. On the left, you can see mice not treated with XJB have a much wider spread of CAG repeats, showing that this region is unstable, as we've seen previously. On the right, mice treated with XJB show a different profile in that the peaks are much tighter and you see less expansion in mice treated with XJB. When we averaged results for groups of mice, we show the graph on the bottom left, we see that treatment with XJB reduces the change in repeat length in these mice between birth and 20 to 30 weeks. So mice treated with XJB expand their repeats up to around 10, whereas mice not treated with XJB can have an increase of up to 35 CAG repeats. Over on the right, we show the motor function tests that we did for mice at age 9 weeks, shown in blue, age 28 weeks, shown in red, and age 57 weeks, shown in green. And we show that mice treated with XJB retained motor function for much longer than untreated mice. At the young ages, the time the mice were able to stay on the rotating rod is around the maximum time of 120 seconds. With age, this time decreases, as could be expected. In the untreated Huntington's mice at age 57 weeks of age, their performance has dropped by over half, whereas in the XJB treated mice, they were able to retain much more motor function and to stay on the rod for much longer. We found similar results in the bar grasp test, with mice that were treated with XJB retaining more motor function at 57 weeks, this is the green bar on the bottom right, than mice not treated with XJB. So this brings us to our model of somatic expansion and potential for changing the spectrum of Huntington's onset. In the inherited model of Huntington's at the top, the inherited expanded gene causes decades of effects from the toxic effects of the expanded polyglutamine protein. This toxicity would build up and cause onset at an age that was determined by the expansion that you inherited. However, in the somatic model, the gene that you inherit continues to expand through life and at some point hits a threshold number of repeats, at which point you get onset of symptoms. Therapeutics that could block this somatic expansion could potentially delay reaching this length threshold and delay the onset of symptoms. So this is a new area for possible therapeutic intervention. So to summarize what I've talked about today and the results of our studies, we showed that blocking somatic expansion can delay onset of Huntington's disease in a mouse model of HD. By knocking out the OGG1 gene and reducing the number of strand breaks, we suppressed somatic expansion and also delayed motor decline. Furthermore, treating with XJB5131, an antioxidant drug that's targeted to the mitochondria, also reduced somatic expansion and delayed loss of motor function in the Huntington's mice. So this is a new avenue for therapeutic treatment for HD. By delaying the onset of symptoms, we can offer hope for people who inherit the gene. So I'd like to thank everyone who was involved in this study. This work was done in the lab of Cynthia McMurray at Lawrence Berkeley National Lab with the help of Paul Williams and Doyup Lee. The mouse testing was conducted at Mayo Clinic with the help of Faye Harris. DNA analysis of the CAG repeats was done by ACGT. The histology for this study was done by Jens Panke. The analysis of the OGG1 and Huntingtin protein in these samples was done by Bartosz Szczesny. Mitochondrial DNA damage was tested by Silvette Ayala-Pena and Karina Acevedo-Torres at the University of Puerto Rico. XJB5131 is synthesized by the group of Peter Wipf at the University of Pittsburgh. This study was funded by the NIH, and I would like to thank HDSA for funding my work. And I would like to thank all of you for your attention, and I will answer any of your questions.
Thank you so much, Helen. That was fantastic. A great overview of your really exciting work. While I'm opening up the questions, I'll just remind everyone that you can just type your question at any time and click send, and we'll make sure that Helen gets them. One question coming in, Helen, is did you observe differences in the degree of somatic expansion in the striatum with respect to other brain regions?
That's a very good question. Yes, we did. So as has been shown before by many groups, the striatum is the region most affected in Huntington's disease, and we did see a greater degree of somatic expansion in the striatum. And by that, what I mean is somatic expansion was measured in 4 brain regions, in the striatum, the cortex, the cerebellum, and the hippocampus. And we saw expansion in all regions, but we saw a different pattern in the regions of the brain. In the striatum, we saw longer expansions and larger repeats. And so we think this is what correlates with that region being the most susceptible.
Okay. Another question, Helen, is this might be a little tough one to answer, but how do mice genes compare to human genes? In other words, if either, I think they're referring to either the cross or the drug, delays mouse onset, I guess a particular amount of time, is it a one-to-one correlation to human delay?
That's an interesting question. So in the mouse, this is a great system where we can study gene knockout. This is a technology that's been used a lot in mice. So by knocking out the OGG1 gene, we were able to study the effects of somatic expansion without causing additional negative effects in the mice. By knocking out this gene, you do get an increased number of oxidized bases, as you would expect since you're not removing them. There are backup systems in the mice, and these bases will be removed, and so at older ages in our study, we actually could not separate the effects of somatic expansion in the OGG1 knockout mouse from the OGG1 containing mouse. Whether it's a one-to-one, that's much more of a speculative idea. A lifespan of a mouse is around perhaps 2 years, so trying to do a one-to-one comparison to a human would be very, very difficult, but it certainly indicates that these treatments could delay onset of symptoms. By exactly how much, since there is such variation in people, it's hard to put a definitive number on, but certainly would likely lead to a delay in onset.
Thank you, Helen. I have a question in regards to the XJB compound. So I'm assuming, or first part of the question is, do you believe it's the free radical scavenging ability of the compound that is at play here that's causing the changes in motor behavior of the mice, or is it some other mechanism that you believe is at play?
So mitochondrial dysfunction has been noted previously for Huntington's disease, and since XJB is targeted directly to the mitochondria, it could be having other functions that are causing this benefit to symptoms of Huntington's disease. We are testing for that. One thing that we looked at in our studies with XJB was mitochondrial copy number, and also the amount of DNA damage in the mitochondrial DNA itself. Mitochondria also contain their own DNA. So these are things that we're looking at. It's certainly possible that there are other effects of XJB going on, and any antioxidant that's mopping up free radicals is likely to have a much wider effect than just on the somatic expansion of the repeats. But these are things that we are testing for.
Okay, you kind of answered part two of my question, was that do you have any evidence in the mice that were given the drug that got better, that there was any change in free radical damage, DNA damage, mitochondrial respiration, things like that, that could correlate with things you're seeing in rotarod behavior? As you're saying that data are pending, or you're looking at them.
Yes, these are things that we are testing.
Does the compound, is there a link between the compound and OGG1? Do you know if that inhibits OGG1?
We don't know of any link. Really, there are two alternative ways of looking at the same thing. In the toxic oxidation cycle model that I put up, if you reduce the amount of oxidative damage, in theory you don't get the oxidized guanine bases, so you would actually take away the thing that OGG1 acts on. As for a direct effect of XJB on OGG1, we haven't looked at. What we believe we're seeing is really just two ways of achieving the same end.
Another question just came in, and this is inquiring about other genes. As people may or may not know, obviously this attendee does, that Lots of other genes have CAG repeat expansions in them, or CAG tracts, and they're asking about what about other genes with CAG tracts? Did you observe expansions in these genes in the HD mice versus the wild-type mice?
So in our studies, we have only looked at the CAG tract in the Huntington gene. What we believe is that this model of somatic expansion actually applies to many of the trinucleotide repeat diseases, and that includes not only the other CAG repeat diseases, but for some other conditions such as Fragile X, which has a CGG repeat, and some of the other trinucleotide diseases. So we believe this is actually a common model of somatic expansion, but in our studies we only looked at CAG repeats in the Huntington gene. Expansion of our work could look at other regions and other trinucleotide repeats.
Great, thank you. How long, for your mouse study with XJB, how long did you have to treat those mice to see the observed effects? Was this a chronic administration of the drug or other?
So these tests are all ongoing, and we're looking at different lengths of time for treatment and different starting times of treatment with XJB, but those mice in that study were treated from young ages all the way through up to beyond 60 weeks. We tested motor function at 9 weeks all the way up to 57 weeks. So that was a chronic dosing of XJB. They were dosed at regular intervals throughout their lives.
Okay. Great. And one more question, Helen. I'm wondering why XJB, I'm sure you looked at maybe other free radical scavengers, and we're just wondering the rationale of why you selected this to move forward first with animal testing versus maybe other free radical scavengers that you may have tested in the lab in vitro or otherwise.
So we know that antioxidants have been trialed in Huntington's disease before with quite mixed results. One of the reasons to use this antioxidant is really for its direct targeting to the mitochondria. We believe that adds a level of specificity that more general non-targeted antioxidants may not have. By being targeted directly to the site of this free radical production, you're able to scavenge these free radicals at their source, which would hopefully minimize the damage before they are released further into the cell to do their damage. So we believe that that's one of the reasons that this could be a better option than some of the more general non-targeted antioxidants.
Do you have evidence in at least the mice that the XJB compound gets into the brain?
So we have done studies, and so this is more the work of Cynthia McMurray, so I don't want to speak out of turn here, but I believe we do have evidence to show that this compound crosses the blood-brain barrier.
Okay, great. And I see we're a few more minutes, but we have one more question, and I know this will be a very difficult one to answer, but is there a timeline for human clinical trials? Is the question coming in from the community. I don't know if you would care to respond to that at all.
What I can say is that XJB is, we're currently in the stage of preclinical trials, so we're still doing mouse studies to confirm that this is safe, that this is effective, to get a really good handle on the dosing, the time and the amount that we dose. This is being pursued by Cynthia McMurray, and so it's very difficult to put a timeline. I know everybody understands that these things do take a very long time, but what I can say is that this is in the preclinical testing phase.
Fair enough. Thank you, Helen, and we appreciate that. We know that, you know, these things take quite a bit of time from conception, from moving from animal studies all the way through clinical testing, and eventually, hopefully, FDA approval. You know, these drugs can to remind the audience, can take anywhere from 12 to 15 years from conception to FDA approval. So it's a long road, but the data are exciting, and this is a very exciting potential pathway, novel pathway of yet, where we may potentially be able to intervene to delay the onset of Huntington's disease. So it's very exciting. So with that, I want to thank Helen for taking the time and walking us through these data and joining us so early in the morning. I really appreciate Helen, and thank you everyone for joining us. Make sure, if you can, to join us on October 7th for our next research webinar at 12 p.m. Eastern. And until then, that's all for now. Have a great day, everyone, and we'll hopefully talk to you soon. Bye-bye.
Thank you.
Bye-bye.